DNA, deoxyribonucleic acid, is a molecule that acts as a blueprint for any living thing. DNA elements combine to form the shape of a long, spiraling ladder or a double helix.
The steps of the ladder in a DNA double helix are made out of 4 different kinds of chemicals, or nitrogenous bases, named thymine, guanine, adenine, and cytosine. These are the fundamental codes that controls all genetics. Thymine only pairs with adenine, and guanine only pairs with cytosine, because A and T will always make a right amount of hydrogen bonds, 2, and G and C will always make 3.
This is also because adenine and guanine have one ring in their structure called purines, and thymine and cytosine have 2 called pyrimidines, and only bases with 2 rings can bond with those with 1. So they can complement each other, fit each other perfectly, and have no molecules left over. DNA's backbone is made up of phosphate and sugar molecule covalently bonded, where phosphate is bonded to sugar, which is bonded to one of the 4 nitrogenous bases. One sugar, one phosphate, and one base molecule combined is called a nucleotide.
DNA replication is the process in which DNA in a cell makes the exact copy of itself during interphase before the cell division for growth, reproduction, and repair.
The semi-conservative replication is the most accurate model of how DNA is copied. This means that in building the new DNA double helix, half of it, one strand, will be old, and the other strand, will be new.
The first step in replicating DNA is separating the old DNA double helix. An enzyme, which is something used to speed up the reaction by the name of helicase, cuts the hydrogen bonds between the bases pair so that the DNA is left with 2 different single strands.
Next, another enzyme called primase goes through each strand and sets down a primer called RNA primase at the beginning of each strand, giving a starting point for the polymerase to begin.
Polymerase is the next step, and it is basically another enzyme that is in charge of finding and collecting the base pairs floating around the nuclei of the cell, and the connection them to their respective base match on 2 single strands of DNA. In this way, the old single strands have been used as templates to create 2 new single strands of DNA with their complementary bases.
Finally, the last enzyme, ligase, comes in and links the small gaps or fragments of newly synthesized DNA with covalent bonding of sugar and phosphate. This process is done twice, one time for each strand. Primase sets the primer, polymerase joins bases, backbone, and H bonds, and ligase closes the gaps.
That would melt in your hands, and she spoke words of wisdom to the basement people, to the basement. Many surprises await you in the basement, people in the basement. You heard that last time, you know we're gonna find you sick in the car, sick 'cause you're not up to going.
And just like that, 2 new double helix strands of DNA have been made.
Sick in the car, sick 'cause you're not